Today we're going to talk about transcription. So we're starting with the central dogma of gene expression. This gives us sort of an overall view of how gene expression works. We're going to start with DNA. Remember, DNA actually contains a complete code for all of the proteins that are going to be made by a person. The question is, how do we get from DNA to protein? Well, the first step that we're going to follow is transcription. We'll talk about that in this video. What happens in transcription is DNA, the code in the DNA, is transcribed into mRNA. I often like to think of this just like the secretaries used to do when they would transcribe their office manager's dictation into words. And that's exactly what we're talking about with transcription. We're not changing the context. We're not changing this from the language or anything. Basically, all we're doing is just taking from one type of nucleic acid to a different kind of nucleic acid, from DNA to mRNA. In the next step, now we will translate the information from mRNA into protein. This is like a completely different language. That's why you can think of it as translation. We're going from nucleic acid to amino acid. Now, in order to understand this, you need to understand that there are three classes of RNAs. The first class of RNA is the messenger RNA. This is what actually carries the protein building instructions, often called mRNA. Then we have the ribosomal RNA, or the rRNA. This is actually what makes up the component of the ribosomes. So the ribosomes themselves are built out of ribosomal RNA. And finally, we have transfer, or tRNA. You can also think of this as translator RNA, because this is the one that actually translates the information found in the mRNA into the amino acids that are built by the ribosomes. So just to remind you, here's a nucleotide subunit of RNA. And remember, the difference between DNA and RNA lies right here in this OH group down at the bottom. Sugar, in this case ribose, that is found in the RNA, has an extra OH group in it. This OH group on the 2' hydroxyl, this extra oxygen involved, is actually what makes this much more reactive and much less stable than DNA. Now, when we're talking about transcription, we have to realize that it looks very much like replication. Essentially, all we're going to do is pair the bases. The difference here is we're going to make a copy of the DNA strand and we're going to copy it into RNA. Now in RNA, U's are not used. We have T's are not used. We have a uracil rather than a thymine. So what happens is, when we have the code in the DNA and we're going to transcribe it into RNA, wherever we would normally have a T, we would fill in a uracil. And so that we get this sequence here. And if you compare this to what we see down here, the matching pairs during replication, you can see an important part of the difference. Transcription and DNA replication in some ways are very much alike. And so it helps to understand them in terms of how they're alike and how they're different. So like DNA replication, the nucleotides are always added from the 5' to the 3' end. Just like we talked about before, where we noted that all of the DNA that's built is always built 5' to 3' as well. So is it with RNA. But unlike DNA replication, we're only going to make a small stretch of this. We're only going to copy a portion of the gene that we're going to use in order to code for the protein. This is going to be coded by RNA polymerase, which is going to catalyze the nucleotide addition, unlike replication, where it was DNA polymerase that was catalyzing nucleotide addition. And the product is going to be a single strand of RNA rather than a double strand as we did during replication. The promoter is where the gene is going to start. This is what signals the RNA polymerase where the gene begins. Remember, it's a huge sequence of DNA, and there's no place in it that specifically tells the polymerase this is the start of a gene. It reminds me of when my brother first got a computer, and on his first computer you actually use cassette tapes in order to load programs. The problem was, on the cassette tape there was no way for the computer to know where the program began. And so what you had to do was you actually had to write down where the counter was on your cassette tape in order to tell the program where it started. This is how old the original computer that I'm talking about is. But the point is exactly the same. Within this entire long stretch of DNA, we need some way to find where the start of the gene is, and that's the promoter. It's simply a base sequence in the DNA that signals the start of a gene. For transcription to occur, the RNA polymerase must first bind to the promoter. And once it binds to the promoter, that identifies the start of the gene, and it can begin making the copy, making the mRNA copy. So here's a picture of what I'm talking about. Here we can see the RNA polymerase has sat down in the promoter region and has identified the start of the gene. Once it identifies the start of the gene, then it's going to begin to transcribe the gene. It's going to start matching up the base pairs. So here we can see what's going on. Now remember, the copy that we're using here, we're actually working off the opposite strand. We're working off the strand that goes 3' to 5' because we have to build 5' to 3', right? So we always have to be using the other strand. So in fact, this strand here is the one that we're going to copy. The RNA transcript begins, and notice here we match the T, we match an A, C we match to a G, and A, notice we match it with a U here in the RNA. And it moves down the line, building along all of this transcript until eventually it gets to a end of the gene. It gets to a stop region. And that stop region will basically cause it to stop producing the RNA transcript. Now, an important thing to realize, the RNA transcript is actually going to be longer than the coding region. There will be parts of this when we go into translation which will signal where the actual gene starts and stops. So this is like those old films where they have a little bit of extra leader region and a little bit of trailer at the end of the film. That's what this is exactly like. So the gene is actually only in the center part. Hopefully this video has helped you. The next video we'll be talking a little bit about translation.